So hello everyone, good evening, how are you all? So guys, today we are going to discuss about heart, and this is one of the most important topic for railways, and also the questions which are asked in the SSC examinations like CGL, CPO, the questions are in the statement wise form. So in order to understand what this question is asking, and in order to take the correct answer, we need to understand this with the basic level. So let's understand what are blood vessels and what are heart. So let me use my plotter here, and first of all, let's have a look. This is the body structure in which the heart is present in the central part of your body. That is, if I talk about the thoracic cavity, so yes, that's present in your thoracic cavity, right? And now here we have blood vessels via which this blood actually circulates. So these are pipe-like structures which are present in our body. So these structures can be like veins, they can be like arteries, right? So first of all, let's understand what is blood. So blood is actually a connective tissue. What is it? That's a fluid connective tissue. And here we have two parts. One is plasma and another one is formed elements. So let's have a discussion over that. So guys, what is plasma? So that's 55% of our blood, and that's the liquid portion of the blood. Fine. So what is it? This is actually the liquid portion of the blood, which is known as plasma. And it is straw colored and slightly alkaline, where pH value is approx 7.4. We can say that is slightly basic in nature, right? But there are 45% of RBC, WBC and platelets which are known as formed elements. So 45 and 55, it will totally complete your blood. Fine. So proceeding further. Now one by one, let's have a discussion on it. So here, if I talk about this plasma, this is the whole blood. So this portion, this yellow colored portion is actually plasma, right? So plasma, in the plasma you have 90 to 92% water, and rest 6 to 8% there are proteins. And what kind of protein? So here we have fibrinogen, globulin and albumin. Fibrinogen, that is used for the blood coagulation, or we can say blood clotting. Globulins act as the antibodies, that is, it acts as a defense system of our body. So in case virus, bacteria, they attack your body, so they are ready to fight against them. So now moving forward to the albumin, so they maintain the osmotic balance and they regulate our blood pressure. Then we have glucose, amino acid, lipids, cholesterol. We have sodium, magnesium ions as well, and then we have gases like CO2, O2 and all. That's for the transportation. One more thing, please remember, this is the serum part. Now what is the difference between plasma and serum? So if clotting factors are not there, right? So plasma without the clotting factor is known as serum, and that's the yellowish part of our blood. Understood? Great. Proceeding further. So guys, first of all, let's talk about RBCs now, because they are the first part of these formed elements. And you must remember that for 55% was actually plasma. That was the liquid portion, right? But if I talk about RBC, WBC, platelets, they are in the part of 45%, in which we'll say RBC is approx 5.5 millions. What is the count? 5.5 millions. So yes, RBCs or erythrocytes, what is the count there? Its count is 5.5 million. And if I'll compare it with the lakhs, so 10 lakhs is equal to 1 million. Okay. Then white blood cells or leukocytes, what is their quantity? So that's approx 10 to 10,000 to 12,000 per microliter. 10,000 to 12,000 per microliter. And similarly, if I talk about platelets, they are known as thrombocytes, but you need to know its quantity, and that's approx 1.5 to 4 lakhs. Now, since we are talking about their quantity, so are they asking the examination? Yes, they are asking the examination. And one of the questions which was asked previous yearly, let me mention that also. The question was, what is the ratio of the quantity of RBC with the WBC? So that ratio is 600:1. So that was asked in the previous year paper. You must remember this. Great. Proceeding further. So now, proceeding further. So what is the role of RBC? So RBC helps in the oxygen transportation. It is formed in the bone marrow. Apart from that, they die in the spleen, and that's why spleen is known as the graveyard of the RBC, right? Okay. So yep. So RBCs, they are red because of the presence of hemoglobin. Normal hemoglobin in the blood is 12 to 16 gram per 100 milliliter. So we link it with the 14% in the males and 12% in the females. Then lifespan is of 120 days, but in the examination, it was asked what is the lifespan, and there that duration was given, and that was 80 to 120 days. You must remember this. that the lifespan of this RBC is 80 to 120 days. Then the count is 5.5 millions per millimeter cube. Then we have it is formed in bone marrows and the structure is biconcave in shape. There is no nucleus, no mitochondria, no Golgi complex. So this is actually asked in the examination. You must remember this. Okay, what are the functions? So it helps in the transportation of the oxygen and carbon dioxide. Proceeding further. So worn out RBCs are actually destroyed in the spleen, and that's why spleen is known as the graveyard of the RBC. We have discussed this. Next. Now as we have WBC, they are colorless. Their lifespan is of 1 to 15 days. Here the count is, as discussed, 6,000 to 8,000 per millimeter cube. Then it is formed in bone marrow, lymph, gland, spleen. Structure is nucleated. That means it has nucleus, but RBC doesn't have nucleus. Do you know what are the exceptions? The exceptions are llama and camel. So in the camel and llama, you will find that nucleus is present in the RBCs, but that's an exceptional case. You must remember that. Fine. So what is it? So that helps in maintaining the immunity. So here we have neutrophils, eosinophils, basophils, lymphocytes, monocytes that's present here. And if I talk about the functioning, so neutrophils, so their functioning is as a soldier of the body, that is phagocytes. Then we have eosinophils, so they resist infections or allergic reactions. Basophils, they secrete histamine, serotonin, heparin. Monocytes, that is phagocytosis. If I talk about lymphocytes, they maintain the immune responses of the body and they secrete antibodies. So guys, this was all about WBCs. Proceeding further with the platelets. So they are colorless, their lifespan is of 7 days and their count is of 1.5 to 4 lakhs. Okay, but guys, do you know what is the case that happens with the dengue? Dengue, when mosquito bites you, but it's not all the mosquito. If there's a virus in that mosquito and that virus is dengue virus, if it bites, so there the number of platelets actually decreases. So what you can have in that case? You can have the milk of goat, right? Goat's milk will work there. You can have papaya. They all are good sources because of which these platelets count can be increased. And what is the role of these platelets? So these platelets are helping in the blood coagulation, right? So that's mentioned here, that helps in the blood clotting. Okay. So what's blood coagulation? So let's say you are playing cricket, you are playing kho-kho, you are playing anything. So if while playing or while doing any task, if your finger gets cut or your skin gets cut, you will get bleeding there, right? But that bleeding stops within 2 or 3 minutes because of this vitamin K, because of this platelet, because of this prothrombin. So they help in blood clotting. And what is it that prevents the blood clotting within your body? That's heparin. Both the questions are important. Fine. So that was about blood coagulation. So if I talk about the blood coagulation steps, so there's a site of injury where you have been hurt, right? Where wound is there. So here you will have the clumped platelets and tissues. They'll release the thromboplastin, which forms the thrombokinase. Here calcium ion is there, that hydrolysis, and here prothrombin is turned into the thrombin. Then fibrinogen, which is soluble in the state, it forms fibrin, and this actually traps the that particular blood part, and yes, finally the clot is formed. So this is the stage in which there was injury, blood was coming out, platelets plug here, fibrin clots, and finally the blood clotting is there. It's as simple as that. The only thing is that we have to try to understand what is being asked in the paper. Everything is simple, but you have to use your brain there. So is there any doubt, any queries regarding same? If everything is sorted, we can proceed further. So guys, yes, these are one of the important things which are repeatedly asked in the examination, but we think that we'll just mug it up and try to take the answer. At that time, issues created. But as of now, I guess this part is sorted. So that was about blood clotting. So it's mentioned here. So these are the platelets, these are the RBCs, and these small dots, these are actually WBCs, which are maintaining your immune system. Okay. So injury that results in the bleeding, and finally here blood clotting is done. And I've already explained you how this clotting was done. Okay. So now guys, let's move forward with the blood grouping. So can I get the answers from you people? Guys, what are your blood groups? So I'll start with my group. My blood group is O positive. So what's your blood group? So guys, when I'm saying O positive, this means that there might be O negative as well. So what's the difference between the positive and negative? Tell me. So guys, if I'll talk about A positive or AB positive also, so let's have a look at that. See, we have 4 types of blood group. It's A, B, AB and O. There are 4 types of blood groups, right? But there was an experiment that was done on a monkeys category that is known as rhesus macaca, and it was found that the protein that was present on that monkey, if this protein is present in your body, that is in your blood on the surface of the RBC, you will be considered as positive. If that protein is not there, you will be considered as negative. As simple as that. So yes, this naming RH is actually derived from rhesus macaca term, that is RH. So RH factor, because of this RH factor, the 4 blood groups are divided into 2 parts each, and hence it becomes total 8 parts. So blood groups, they are just in 4 categories, but because of RH factor, it's A positive, A negative, B positive, B negative, AB positive, AB negative, and O positive, O negative. Right? Perfect. So guys, here blood groups were discovered by Karl Landsteiner, which includes ABO grouping and RH grouping. Okay. So if I talk about this grouping, so ABO grouping, see, here we have A antigen but B antibodies. Here we have antigen B but A antibodies. Here we have AB antigens, no antibodies. And here we have neither of the antigen but both the antibodies. So yes, I have a chart for you all and you must remember this. And if you want, I have an animation also to explain you. First of all, just have a look here. So guys, these are the blood groups. Now, always remember the antigens or antibodies, what are they? They are basically the proteins, right? Okay. So either it will be A or it will be B. There are no other option. Okay. Now, guys, if there is blood group A, the antigen is actually A only, but antibodies are just opposite. So the blood group A will have antigen A but antibody B. Blood group B will have antigen B but antibody A. AB will have both antigens but no antibodies. O will have no antigen but both the antibodies. Here, antibody decides the blood can be donated or not. Just see, because in the blood group B antibody was there, so it keeps a check. Oh, the person with B, you are not allowed, otherwise I'll shoot you. So if A is in the need of blood, A can give the blood, no worries. But this B is acting like a security guard. It keeps a check. B is not allowed. So B's entry is denied. Again, you will find B here. So for the A, if blood is required, only and only blood can be gained from O, A and O. Similarly, if blood group B needs the blood, who can donate the blood? Just check. Antibody A is present. So A can donate the blood? No, not allowed. B can donate the blood. But can AB donate the blood? No, again, A is there, cannot. So guys, here, if blood group B needs blood, B and O can donate the blood. Let's talk about AB. So AB has any antibody? No sir, there is no such antibody. That means there is no such security guard. Everyone can donate the blood. So yes, AB can receive the blood from A, B, AB and O. And hence we can say that AB is the universal acceptor. What about O? O has antibody A also, antibody B as well. So neither A nor B nor AB, no one can donate the blood. Yes, O can get the blood from O only. So you will find that O is donating the blood to everyone. So O is known as universal donor and AB is accepting the blood from everyone. So AB is known as universal recipient. As simple as that. Got it? Perfect. Next. So proceeding further. So if I talk about this ABO grouping here, just see. So antigen A reacts with antibody A, antigen B reacts with the antibody B. So mixing of the blood will with interactive antigens and antibodies causes clumping, that is agglutination of the RBCs, or we can link it with the blood coagulation, or we can say blood clotting is there, right? Okay. So now proceeding further. So O group is known as universal donor because O can donate the blood to everyone, and AB is known as universal recipient because it can gain the blood from everyone. So the questions when it will be asked in the statement wise form, you can easily attend that. So that's done from my end. Now RH grouping. So what was positive and negative? So yes, these were the proteins that were present in the monkeys category, that was rhesus macaca. So if this protein is present, obviously the person will be positive. If this protein is absent on the RBC, then negative. That's what is mentioned here. Rhesus, that is RH factor, is an antigen found on RBC. RH positive means the presence of RH factor. RH negative means the absence of RH factor. So nearly 80% of the humans are Rh positive. Maximum of us are positive, and that's why I asked in the comment section, can you please tell me what is your blood type? In our class also, I can take this guarantee because I've been teaching from past 10 years with my selection. I have given selection to the candidates also. My selection is also there. So this is all from experience, and yes, obviously this is there in the articles as well. Most of us in our class are positive only, and that too you'll surely get O positive as a very common group. Okay. So talking about this RH grouping, see, if there's a RH positive female, if there is RH positive male, with their combination, Rh negative child cannot be there. That's what it was mentioned. But yes, if Rh negative mother is there or Rh positive father is there, so Rh negative property can be inherited from the parents. So actually, it all depends upon the parents because the genes are actually transferred from the parents only. So everything depends on our father and mother. So half of the chromosomes are gained from father, half of the chromosomes are gained from mother, and then we'll get the blood, obviously, right? So anti-Rh antibodies are not naturally found. So Rh negative person can receive Rh positive blood only once, but it causes the development of anti-Rh antibodies in his blood. So a second transfusion of the Rh positive blood causes agglutination. Therefore, Rh negative blood group should be missed before transfusion. And what I'm talking about here? Here we are talking about erythroblastosis fetus. What is that? So if Rh negative mother is pregnant with Rh negative baby, at that time the first baby will be fine, but from the second baby itself there will be issues like severe anemia or severe jaundice will be there, because of which there might be the issue in which even the death of the baby might take place. But that is the case when mother is negative, when mother is Rh negative and the baby is Rh positive. So the condition was mentioned here. Okay, so here what is erythroblastosis fetus as discussed. So this is a pregnant mother where the mother is Rh negative and the baby is Rh positive. So in this condition, it's mentioned here, it is Rh incompatibility between the Rh negative blood of a pregnant mother and Rh positive blood of the fetus. So Rh antigens do not mix with the maternal blood in the first pregnancy because of the placenta separates the two bloods. Okay. So, but at the time of the first delivery, there is a possibility of the exposure of the maternal blood to a small amount of the Rh positive blood from the fetus. This induces the formation of Rh antibodies in the maternal blood. Fine. Okay. So, in case of her subsequent pregnancies, Rh antibodies from mother leak into the fetal blood and destroys the fetal RBC. This is a fatal to fetus or causes severe anemia and jaundice as discussed, and this condition is known as erythroblastosis fetus. That's the condition. Okay, so this is again done. So here erythroblastosis fetus can be avoided by administering anti-Rh antibodies to the mother immediately after the delivery of the first child. So you can just see the color of the baby is yellow, right? So we can link it with the jaundice, and in Hindi, in the North Indian belt, we call it pelia because the structure of the body is actually yellowish in color. Okay, so that's done. So if I'll talk about the types of blood vessels, they are arteries, veins, and capillaries. So that's about the blood vascular system. So blood vessels, they may be arteries, veins, or capillaries. So arteries are the ones that carries the the oxygenated blood. That is the blood with the max amount of oxygen. Veins are the ones that carries the blood with the lesser amount of oxygen, where carbon dioxide content is more. And capillaries are the ones in which the blood transfusion is the mixture of the blood is taking place. Okay, so arteries, as discussed, are the ones from which the blood with the oxygen is traveling. Veins are the ones from which the blood with the carbon dioxide maximum content blood is flowing, and these are the capillaries, and you can find these capillaries in your eyes. You can just open your eyes and look at in the mirror. You will find small red like line like structure. They are basically the capillaries. And when you measure the blood pressure, so which part is used? So that time we are using arteries. So the device that is used for measuring the blood pressure, that is sphygmomanometer, and where it measures the blood, so that is measured on the arteries part only. Understood? So I guess now it's clear. So this capillary is basically the junction as discussed. Arteries are the ones from which oxygenated blood flows, and veins are the ones from which deoxygenated blood flows. So I guess this has given you the clarity. And guys, if you have any doubts or queries, you can just ask. Do not hesitate, because these things are very important and very relevant with the examination. Okay. So they carry the blood from the heart to other tissues. They contain the oxygenated blood. except the pulmonary arteries. So we are talking about this blood vessel, especially the arteries, and their smaller branches are called arterioles. Fine. Okay. What about proceeding further? What about veins? So veins are the ones that carries blood towards the heart. They contain deoxygenated blood except the pulmonary vein, and their small branches are called venules. So veins also have three layers, but tunica media is comparatively thin. So it's mentioned here. Done. Now proceeding further, capillaries were the ones that were just acting like a junction point. So here we have arteries and here we have veins. That part is known as capillary. I guess this part is sorted. Okay. So we have discussed about all those things. Now is the time to understand the structure of heart. So guys, this is the heart which is present in the thoracic cavity, and guys, it's slightly tilted in the left. That's why we feel that lumped up sound on the left hand side, right? Okay, so heart is mesodermally derived organ which is located in the mediastinum. It is protected by the double layered pericardium, and there's a fluid that is present here, and that is known as pericardial fluid that gives the lubricacy to the heart. Fine. So I'll just show you here. This heart has four chambers where we have two upper chambers that are known as auricles and two lower chambers that are known as ventricles. So it's like this. You must remember this. So guys, heart is divided into four chambers. This is the right portion, this is the left portion. So right auricle, right ventricle, it's left auricle, and that's left ventricle, right? So these are the four chambers of heart that is present in our body. So if I talk about humans, so yes, here we have four chambers. Okay, then so atria is separated by the interatrial septum. So ventricles are separated by the interventricular septum. In short, septum is that part which is separating the left and right portions of the heart. That's asked in the examination, so you must remember this. Okay, so next. So now the valves, how they are functioning. So let's have a look. So these are the conducting systems. 1 second. I'll just directly show you the cardiac cycle. So guys, that's the heart, right? We were talking about the heart, right? So this is our heart. Now within the heart, these are those pipe-like structures which we were talking about, from which the blood will come and it will move throughout the body, right? So these are the four chambers of heart that I was talking about. So right atrium, right ventricle, left atrium, left ventricle, and within two compartments, that is within the heart. If I talk about the atrium and ventricle, so these are, there we have what? We have structures which are preventing the backflow. What is that? That's known as valve. So in the right atrium, we have tricuspid valve. On the left part, we have mitral valve. So you must understand this bicuspid valve, tricuspid valve, mitral valve, where they are present, they are asked in the examination. So yes, if I talk about the phases of the cardiac cycle, so we have joint diastole, atrial systole, and ventricular systole. So what is it? So in order to understand this, see, I was talking about these arteries. Sorry, these flap-like structures which were preventing the backflow. These are the valves. Okay, so it is the cardiac cyclic contraction relaxation of the heart for pumping the blood. Now guys, if I need to check the blood pressure, what device I'll use? I'll use sphygmomanometer, right? So sphygmomanometer is used to measure the blood pressure. So this, what is the normal value of this blood pressure? You must remember the value of normal blood pressure is 120/80. What is it? 120/80, where 120 is systolic and 80 is diastolic. So this is the systolic and diastolic reading. Now, if the value is more than this, so we will say that the person is suffering from the high blood pressure. But if the value is lower than this, then we will say that person is suffering from the low blood pressure. But what is the name of the device? So the device is basically sphygmomanometer. What is it? Sphygmomanometer is the device. Understood? Okay. So guys, that was all from our heart chapter. So from past 3 days, we have seen the theoretical section, in which on the Tuesday at 5 p.m. we have discussed about the cell. In the next lecture, we discuss about the nutrients, that is vitamins and minerals. And today we have discussed the heart section, in which we have understood arteries, veins, capillaries, then this blood circulatory system. So now how questions are framed. So be ready tomorrow at 5 p.m. So I'll discuss all the questions which are related from these particular chapters, and we'll see how questions are framed in our examination and are we ready for the examination. So for that, do not miss our class tomorrow at 5 p.m. and you can join me daily from Monday to Friday at sharp 5 p.m. Monday we'll be having live test. So on Monday we have live test, right? From Tuesday, we have Tuesday, Wednesday, Thursday as the topic classes, and on the Friday, I am providing you free ebook, that is free ebook, so that you can actually revise the most important questions and most important stuff from our examination. And in this way, we are completing the whole GS portion. So please let me know in the comment section how was this class. Should I continue this series? And one more thing, if you want to study like this in our paid classes, what you can do? You can just go to the Google. So on the Google, you will type Oliveboard. So the first link that opens, you'll just click on it and you will see that there is SSC plus Railway Super Elite batch. Now here, this batch includes both the English and Hindi medium courses. Here we'll have live courses, tests and notes. Everything is present here. So you just need to click on it, and here you'll find enroll now option. Just click on it, and here for you, sale is there. You can use the code JFY to get the 45% off and also 4 months extra validity. So the code for the maximum discount is JFY. So that's all from my end. We'll meet in the next class then. Meet tomorrow sharp at 5, where we are going to discuss the most important the most important questions and the previous year papers related to cell chapter, then your vitamins and minerals, and finally the heart chapter, that is blood circulatory system. So that's all. Please take care, and for the examination, all the very best. Please take care, guys.